In this lecture, we're going to look at microbial genetics or bacterial genetics. Now, a large part of the history of genetics and the current molecular genetics has been elucidated using bacteria and their viruses. So, although bacteria have genes which are composed of DNA, which are arranged in a long series of chromosomes, their genetic material is not organized in the same way as our chromosomes, as our genome. And the reason for that is because they are prokaryotes. And prokaryotes have a different way of arranging their chromosomes. Viruses are also very different from the organisms that you've studied so far. Now, viruses share some of the properties of the organisms. For example, their genetic material is either DNA or RNA, and they usually have a short chromosome. Now, most biologists regard viruses as non-living because they cannot grow or multiply alone. So to reproduce, they must parasitize on living cells and then use the molecular machinery that these cells possess in order to make new viruses. So the viruses that parasitize on bacteria, we call these bacteriophages or simply phages. Now, when scientists began studying bacteria and phages, they were naturally very curious about the hereditary systems. Clearly, it seems that they must have some sort of hereditary system where parents can actually transfer their genetic material to their daughter cells. Now, since bacteria are unicellular, they normally reproduce asexually, and usually this is by binary fission. But scientists wondered, is there ever a union of different types for the purpose of sexual reproduction? In this video, what we look at is we look at how the genetics of bacteria and some viruses, how do they work, and we're going to talk about a few ways by which bacteria transfer genetic material. So the very first way by which bacteria transfer their genetic material to their offspring, to their daughter cells, is by what is known as vertical transmission or vertical gene transfer. Now, vertical gene transfer means that a parent will give its genes to its offspring. So a bacterium, when it reproduces asexually by binary fission, it becomes two cells from just a single cell, and both of these cells are identical to each other, meaning that they contain exactly the same genes which the parent has, which the mother cell has. But there is another way by which bacteria particularly can share their genes, and this is known as horizontal gene transfer or HGT. Now, horizontal gene transfer means that genes can transfer from one bacterial cell to another bacterial cell, even though they are not related in any way. They don't share a mother and daughter cell relationship. Rather, they are peers of each other. So you can call this peer gene transfer from one peer to another. Now, how does this happen? This happens using three different processes. We'll talk about each one of these. Now, when scientists performed what is known as comparative genomic analysis of very closely related microbes, they found that they had different phenotypes, different traits, and this seems to reveal that they have distinct genome sequences or genome differences. And very, very often these differences result as a result of horizontal gene transfer or HGT. It's also known as lateral gene transfer in some textbooks if you look. This means the movement of genes between cells that are not direct descendants of one another. So horizontal gene transfer allows cells to very quickly acquire some new characteristics, and it also causes metabolic diversity to increase. Now, there are three mechanisms of genetic exchange which can be considered to be horizontal gene transfer, and these are known in bacteria. So the first one, like I said earlier, is transformation, in which you have free DNA being released from one cell and is then taken up by another. The second is called transduction, in which the DNA transfer is mediated by a virus. And the third is known as conjugation, in which DNA transfer requires a direct cell-to-cell contact and a conjugative plasmid in the donor cell. So we will talk about these three in detail. Now, these processes are different from one another, and you will notice that the DNA transfer is usually in one direction, that is from a donor cell. So the donor cell is what transfers the genetic material, and it transfers this to another cell known as the recipient cell. So you have a donor recipient relationship between them. It's not a mother and daughter cell relationship, it's a relationship between donor and recipient. Now, before we discuss these mechanisms of transfer, let's just very quickly try to understand what happens when DNA enters the cell by horizontal gene transfer, either by transformation or by transduction or by conjugation. What happens when DNA transfers from one cell to another? So we have three possible fates. The first one is that it may be degraded by the recipient cell's restriction enzymes or some other DNA destruction systems. Number two, it may replicate itself, but of course, this would only happen if it possesses its own origin of replication, such as a plasmid or a phage genome. And third, it may recombine with the recipient cell's chromosome. Now, the word recombine comes from recombination. Now, recombination is actually the physical exchange of DNA between genetic elements. So, structures that can carry genetic information, these are called genetic elements. So, a gene, for instance, is a genetic element. So, one of the ways by which this happens is by a process known as homologous recombination. This is a process that results in genetic exchange between homologous DNA sequences from two different sources. So, what's a homologous DNA sequence? A homologous DNA sequence is a sequence, or rather, homologous DNA sequences are those that have nearly the same sequence. They may have slight differences here and there, and because these differences are very slight, the bases can then pair over an extended length of the two DNA molecules in order to facilitate exchange. Now, this type of recombination, which drives the process of crossing over in classical genetics, for instance. Now, in this lecture, we are not going to look at genetic recombination. It's actually not there in your syllabus. So we will just briefly look at what recombination is, just so that we understand what really happens there. So this diagram here shows you a case where genetic recombination or homologous recombination takes place, and it results in exchange of genetic material between these two chromosomes. So here we have two chromosomes, or rather two segments of DNA, the one which is in light green and the one which is in dark green. And what happens here is we see that there is exchange of their sequences. So some sequences from the dark green chromosome, which is known as the recipient DNA, will then go and attach itself to the light green DNA, which is known as the donor DNA. And because of that, we get these recombination of genetic elements and a recombination of genetic traits as well. So this is one process by which genetic diversity is fueled. Now, there was a question which came, I think, a while back, and this question was, how is genetic diversity in bacteria generated? That was the question, and the answer to this is basically by one of the three lateral gene transfers or horizontal gene transfers. So transformation plays a role in fueling genetic diversity. Transduction also does the same thing, as does conjugation. So these are the three main methods by which genetic diversity in bacteria is generated, and it's for this reason that we see different types of bacteria with different types of abilities, and even within the same species you have a lot of genetic diversity. And it's because of that that we have different strains of bacteria. So one strain can perform a certain activity, and another strain which belongs to the same species cannot. So we will start with transformation. So transformation is a genetic process by which free DNA is incorporated into a recipient cell and brings about some genetic change. Several organisms are naturally transformable. There are some species of gram-negative and some species of gram-positive bacteria which can transform themselves. And in fact, there are some species of archaea as well which can undergo transformation. Now, because the DNA in prokaryotic cells is present as a large single molecule, when a cell is gently lysed, its DNA pours out. Now, bacterial chromosomes are usually very fragile, and because of that, they break very easily. And the reason for why they break is because of their extreme length. For example, if you take Bacillus subtilis, which is a gram-positive bacterium, and you take its chromosome, it would be at least 1700 microns long. Now consider that the typical Bacillus subtilis cell is only about 4 to 10 micrometers long, and its diameter is only 0.25 to 1 microns in diameter, and it's a total cell volume of about 4.6 femtoliters. Now, the unit femto means 10 to the power minus 15. So that's a very, very small volume. So because of its extreme length, even when you gently extract the chromosome of the Bacillus subtilis, which is about 4.2 megabase pairs, you normally get fragmentation. And each fragment is about 10 kilobase pairs, 10 kb. And because an average gene contains about 1000 nucleotides, each fragment of the Bacillus subtilis DNA could potentially contain about 10 genes. Now I say potentially because not all genes are the same length. The average is about 100 nucleotides, but there are some genes which are longer than that, and there are some which are shorter than that. And because of that, the average length is about 1000 nucleotides, which is equal to 1 kilobase pair. Now, this is a size which is transformable. So usually if you have about 10 kilobase pairs in a length of DNA, that DNA length is very good for transformation. If it's longer than this, it might fail. If it's shorter than this, it may also fail. And the reason for that is because it seems that evolution has designed these cells to operate that way. Now, a single cell typically incorporates only one or at the most only a few DNA fragments. So only a small proportion of the genes of one cell can be transferred to another in a single transformation event. Now, whenever we talk about transformation, we always talk about a word which is known as competence or competence. Now, a cell that is able to take up DNA and be transformed is said to be competent, and this capacity is genetically determined. Now, competence in most naturally transformable bacteria is regulated, and there are special proteins which play a role in the uptake and in the processing of the DNA. Now, these competence specific proteins include a membrane associated DNA binding protein, a cell wall autolysin, and various nucleases. One pathway of natural competence in Bacillus subtilis, which is a very easily transformed species, is regulated by something known as quorum sensing. Now, quorum sensing is a regulatory system which responds to cell density. So if you have a certain density of cells, then Bacillus subtilis will be transformed. If it's less than that, it may not. So this is known as a quorum sensing regulatory system. Now, cells also produce and excrete a small peptide during the growth, and the accumulation of this peptide at very high concentration induces the cells to become naturally competent. So Bacillus subtilis cells are naturally competent because of this peptide. Now, one thing you should note, that even though we said that the cells will become competent or competent, not all the cells in a population become competent. In Bacillus, for instance, only about 20% of the cells become competent, and they will stay that way for several hours. Now, if you contrast that to something like Streptococcus, in Streptococcus, you can get 100% of the cells becoming competent, but of course, only for a very brief period during the growth cycle of Streptococcus. Now, there are many layers of regulation which control natural competence in other bacteria. For example, in Vibrio cholerae, the causative agent of cholera, which is naturally found in marine and freshwater environments, and which is associated with crustacean exoskeletons. So, for example, these Vibrio cholerae cells can cause cholera if you've eaten some seafood which has not been properly processed. Now, the competence in Vibrio cholerae is controlled not only by quorum sensing but also by something known as chitin sensing. Now, the exoskeletons of crustaceans contain chitin. Now, Vibrio cholerae can catabolize chitin in the marine environment, and as the cells start to aggregate on the chitin surface, they are in close proximity to one another, and they are more likely to successfully exchange DNA between themselves. So what's going on there is cells which are peers of each other are transferring DNA from one cell to another cell, and because of that we know this as horizontal gene transfer. Let me just remind you one more time that transfer of cells from parents to offspring is known as vertical gene transfer. So high efficiency natural transformation is very rare among bacteria. For example, Acinetobacter, Bacillus, Streptococcus, Haemophilus, Neisseria, and Thermus are naturally competent, and they are very easy to transform. Now, this is known as natural competence, and it provides a nutritional advantage, as free DNA is very rich in carbon, in nitrogen, and in phosphorus. By contrast, many bacteria are very poorly transformed, if at all, under natural conditions. For example, our favorite bacterium, E. coli, Escherichia coli, and many other gram-negative bacteria fall into this category where they are very poorly transformed. However, if cells of Escherichia coli are treated with very high concentrations of calcium ions and then chilled, they become adequately competent, so they can actually take up DNA, although not at a very high efficiency. Now, cells which have been treated this manner can take up double-stranded DNA, and then therefore they can get transformed. So you can transform E. coli even by plasmid DNA once you treat the cells this way. Now, this is very important, this particular topic which we are discussing right now. For example, how do we get E. coli cells to be naturally transformed? Now, in genetics and in gene cloning, you've learned that E. coli are good host cells. They can act as host cells and they can take up DNA. And one way by which we can make them take up DNA is by treating them with calcium chloride and then chilling them. Now, there's another way by which you can make cells artificially competent, and that is known as electroporation. And this is a physical technique that is used to get DNA into organisms that are difficult or sometimes impossible to transform, especially cells that contain thick walls, thick cell walls. In electroporation, the cells are mixed with DNA and then exposed to a very brief but very high voltage electrical pulse. And this makes the cell membrane permeable. It makes the cell envelope permeable. So even the cell wall becomes slightly permeable. And of course, once it becomes permeable, it will allow the DNA to enter the cell. Now, electroporation works for getting free DNA into most types of cells, including our favorite bacterium, E. coli. And even some yeast plants, and some plant cells as well, can respond to this. So you can actually inject DNA into yeast cells and into plant cells if you've performed electroporation on them. So those are the two main methods by which we get cells to become naturally transformable, the so-called competent cells.